Now the third type of grafts possible are the allografts. Allografts are less successful compared to the autograft and isograft. The reason is because they are taken from the same species of the but a different individual, and that means there is going to be genetical difference between the organ taken and the person who is receiving it. The reason is why they are less successful is because the species are going to be same. You are not taking it from some other species, but you are taking from another individual. That individual is not identical twin. So because the individual could be brother in the same family but not identical brother, or a neighbor, or maybe a friend, or maybe a very poor person who is willing to give the organ to a rich person who is dying. In such situation, the two individuals involved are genetically different individuals of same species. That's why chances of rejection are higher. So reason is genetical difference. Because genetical makeup is different, body is able to understand that it is a non-self type of component undergoing transplantation. And the fourth type of grafts possible are the xenograft. Now xeno word is used for foreigner, allo is used for neighbor, xeno is used for foreigner. So whenever a graft is taken from some other species, that means a human taking a graft from a non-human source, in that case it is called as the xenograft. And xenograft are the least successful type of graft, because the body is able to understand very fast that the transplant made is from a non-human type of source. So remember that the cell-mediated immunity is able to recognize the proteins which are involved in the transplanted organ or tissue. That the cell-mediated immunity is able, the T cells are able to understand that the organ taken has the proteins which are foreign in origin and starts fighting against that organ. So if the fight starts, then this organ would be rejected by the receiver's body. That's why what we try to do is we try to bring the immune system down, or to bring the cell mediated immune system down, or to say that the T cells numbers are brought down. So to suppress this response, immunosuppressants are given, and the best example is cyclosporin A, which is obtained from the Trichoderma polysporum. Now what does a immunosuppressant does? So the suppressing agent will destroy the T cell mediated immune response, but it will not destroy the B cell mediated immune response. That means body will still continue to have some amount of immunity or immune power, but the major thing is that the cell mediated immune system has been hampered, so that the body does not reject the organ and the person continues to survive. Now in this concept there is something which you need to remember, that tissue matching, blood group matching, these are very much essential before any type of transplantation or grafting is done. Transplantation may be rejected, and the rejection is already, I have explained, it is because of the immune system of the body. So to suppress the immune system, the histocompatibility agent play, and to suppress the immune response during transplantation, histocompatibility, antigen and immunosuppressants play an important role. Now along with the immunosuppressant, there is something called as histocompatibility antigen. Let's understand this property. So the histocompatibility is the property of having the same or almost the same type of alleles of a set of genes, and these genes are called as a part of major histocompatibility complex. Major means they form a good amount of group, and histocompatibility means matching of the tissue. The major histocompatibility complex, which is short form called as MHC, this gene is basically found on the chromosome number 6. So chromosome number 6 carries a large number of genes, and those genes are a part of MHC, that means major histocompatibility. Now, these genes code for various types of antigen, and those antigens, if are similar, then the body will not reject. If they are different, the body is going to reject the graft. Major histocompatibility complex is also called as HLA, that means human leukocyte antigen system in the human body. So there are set of molecules on the surface of the cells responsible for the lymphocyte recognition and antigen presentation. So this concept play an important role in rejection or acceptance of the graft which is performed on somebody. Now the next topic to start with is the cancer. Now cancer, we all know, is a abnormal cell division which even has uncontrolled cell division, and in this cell division the cell continues to divide, but there is no differentiation followed by the growth. That means the mass or the volume of the cells continue to increase but without any differentiation. And sometimes what happens is that this growth, which is uncontrolled abnormal growth, will start intruding inside the surrounding tissues also. So it starts spoiling and destroying the surrounding tissues as well. The cancer word comes from onco, that's why the study of cancer is called as oncology, sometimes also called as the carcinoma. The word will be coming later in the notes. So the oncology means study of the cancer, and the property of the cancerous cell to move into the surrounding tissue is called as metastasis. Metastasis also is called as secondary growth, and this happens because there are some cancerous cells which are able to break off from the tissue and then spread throughout the body into different portions through blood and lymph. This movement is called as metastasis. Cancerous cells do not show the property of contact inhibition, and they also are able to perform uncontrolled growth and develop into tumorous growth.